If you are not the speaker, we kindly ask you to close your microphone and camera. In addition, if you have some questions, use the chat to pose them. At the end of the presentation, we'll have some time to answer those questions, where our invited speaker is gonna participate with us. Finally, in the chat, we will send you the link to an opinion pool that we kindly ask you to respond. Your ideas and suggestions will help us to improve this experience. Now, on behalf of Tecnológico de Monterrey, School of Medicine and Health Sciences, Tech Salud, we give you the warmest welcome to the first session of Season 5 of the Cátedra Magistral Profesores Destacados, a sharing space dedicated to our students and healthcare professionals, with contributions from outstanding faculty members, as well as recognized international invited speakers. To start, I would like to introduce the moderator of this session. Dr. Rocío Ortiz López is a researcher professor at the School of Medicine and Health Sciences, Tech Salud, as well as the Institute of Obesity Research. Now, I give the floor to Rocío Ortiz to present us the title for this talk and especially to introduce our invited speaker. Welcome, Dr. Rocío.
Good afternoon to everyone. You are welcome to our Cátedra Magistral de Profesores Distinguidos. And this time, we are very, very glad to having here Dr. Federica Di Palma. I will do a very summary here about our guest, Dr. Federica, who holds a PhD in immunogenetics from the University of Reading and brings over 15 years of experience in genomic data science. She has strong international connections in the US through institutions such as NIH, Harvard, and MIT. Dr. Di Palma leads numerous multidisciplinary international research programs for development assistance committee countries. She aims at generating strategic partnerships, building human and infrastructure capacity, and translating scientific knowledge into policies and private sector engagement to deliver long-term impact. Federica Di Palma's previous experience include her roles as assistant director for vertebrate genome biology at the Broad Institute of Harvard, a senior research scientist and service center manager at the Hubbard Center for Genome Studies at the University of New Hampshire, Durham, and a Fogarty Fellow for the National Institute of Health. Most recently, she was Director of Science at the Earham Institute in the United Kingdom, and currently she remains Professoral Fellow in Biodiversity in the School of Biological Science at the University of East Anglia, and Chief Scientific Officer and Vice President, Research and Innovation. So we are giving a warm welcome to Dr. Di Palma, and we are very glad to having you here and all of you. Welcome, Doctor.
Thank you very much, Rocío, and everybody else for this invitation to present here at this seminar today. Can you hear me? Okay, so I'll share my presentation. Is this in presentation mode for you now?
Yes.
Yes. Okay. So thank you very much for the wonderful introduction. That makes it easier for me. I don't have to repeat since you've done it for me already. I wear three different hats, and one is as a Chief Scientific Officer in Vancouver, Canada, and I'll tell you a little bit about what I do there. Another one as a professor at the university, Professoral Fellow in Biodiversity at the University of East Anglia. And I'm also the International Scientific Chair for a global initiative called the Earth Biogenome Project, which is run by Aris Lewin and colleagues out of UC Davis in the US. And it's a global initiative with membership from many, many different countries. At Genome BC, I am the Chief Scientific Officer leading the genomics and science and innovation for the benefit of the Canadian citizen, and in particularly for the British Columbia The mission of Genome BC is that to impact positively lives of Canadian citizens, and in particular in British Columbia, through genomics. We fund a lot of genomics program and a lot of genomics research, and maintain a very close connection with much of the scientific community, both nationally and internationally in Canada, on a number of different sectors: human health, food security, environmental stewardship, and of late, a lot of work on climate change and adaptation, mitigation and adaptation. The vision for Genome BC is to, as I said, impact positively life for every day, and applying the power of genomics to societal and economic challenges. We have many sectors that we apply genomics and technological innovation, and that we fund. We in close collaboration with the scientific community. Some of these sectors are represented here and are very important to Canada as a very natural resource rich country. So far to date, we have funded many projects, over 498 projects, many partnerships in 42 different countries, and received co-investments as well as part of these funding programs. Helped the scientific community to publish many scientific papers and deliver patent applications, and also interacted with a number of companies. This is a geographical representation of many of the centers that work, that are affiliate centers to Genome Canada. And we are here in the West representing British Columbia and called Genome BC. So I'd just like to tell you a little bit now, a bit about my trajectory, my career, my interest, and what brought me here today. And it's mostly, I've always been interested since the very beginning about genomes. I was lucky because I was training at a time, taking my PhD and then doing my postdoc at a time where genome sequencing was becoming available and was being really there was set up and developed through what we have known over the last 20 years of a genomic revolution. And I've been always interested not only in our human genome, but also how we can learn about human health and how we can benefit human health through the genomes of other organisms. In particular, I've been interested in the variability that exists among the organisms within species and within populations. And through my career and trajectory, I then became interested in more dynamic ecosystems, and applying also the knowledge of that diversity outside of the single organism and the one species, but also applying it to an entire ecosystem, as well as understanding later on over the last probably 5 years, understanding how my science and genomics could also have impact and benefit through socioeconomic growth, and how it could have importance for the goods and services that nature provides. So considering really a holistic approach. Of course, I graduated with a degree in cell and molecular biology. And for me, when I started joining the genomic revolution very earlier on, has always been very important, and I trained from the very beginning to really join up two major fields of study, which is the big data, which were emerging from genomics, and the biology, the experimental biology side that I started with. And over the course of my career, I certainly really took a lot of motivation and found very important to apply technological innovation and research excellence to solutions for the sustainable development goal. And this is, of course, as we all know, become a priority now globally. My research goals mostly involves I've always involved pursuing quite innovative and high impact science in very open collaborative environment. That's how I have trained, and that's how I thrive, and I get most of my motivation for science. Answering with many data ambitious questions and really trying to transform a biology by improving, however, the environment and the health. And also, of late, I have reduced my time. As you know, I'm now a chief scientific officer at Genome BC, and only 30% as a professor reduced my time to do research, but I have increased through my new appointments over the last few years, much more an interest in enabling wider national and international bioscience communities. So my interest in terms of research has always been about the understanding of what genetic diversity means and how natural and artificial selection have contributed to the architecture of that genetic diversity, how it has contributed to disease, but how it has also contributed to the evolution of traits that are of economic and ecological importance. Oops. Molecular diversity, of course, understanding also specialized metabolism and understanding how from such diversity one can predict functions from the genomic data, and organismal diversity as well. Looking at species variation, looking at adaptation, looking at the importance of species interactions, co-evolution. And this has led me to understanding, invasive species, but also understanding the connections between the environment and health, and understanding zoonosis, which we know has been particularly important over the last few years with the pandemic. But one of the things that I've always found, perhaps later on in my career, not so much towards the beginning when I was training, of great importance is capacity building, to go really in parallel with the research, fundamental research that I did in my lab. And so of late, I have had the privilege of leading many international development programs in East Africa and in Latin America, and particularly in Colombia. And much of my priority has been to build capacity at the same time as doing research, particularly in situ, in the country. And that has been very much of around genomics methods, informatics, and infrastructure. All of this, however, never forgetting that there is a bigger picture outside of the single biological question, and that is of our benefits, our health. How do we inform a healthy human society within a sustainable environment? And so it's been very important to do science in a very holistic way and interdisciplinary. So if I can tell you a little bit how it started for me, it started obviously learning about the genome, and I was doing, my degree in cell and molecular biology. And so learning about DNA and the book of instructions, the letters that are then translated into messenger RNA, and that the sequence of letters gives us the instructions and the blueprint really of life and everything that we know. I've been particularly interested from the very beginning in the concept of natural selection and evolution, and particularly, variation, and how genetic variation within a population is inherited, and how, for example, there is such things as environment and can lead to competition for survival, and how individuals which have beneficial adaptations are more likely to survive to pass their genes. And over many generations, there is, of course, a change in allele frequencies and all of the understanding of how evolution drives this. So as I said to you, my first important choice is as you can tell from my accent, I am Italian by birth. I was born and raised in Rome, but I went to get a bachelor's degree from the UK, the University of Essex, in cell and molecular biology. I had two options, and one was to be in the field and now they want to be in the lab. And I think because it was the UK and I was from Rome and it was always raining, it was pretty easy for me to decide that the lab was where I wanted to be. And I later on continued to study genes, but I took my PhD in immunogenetics, which was the application of genetics to the immune system. And that was something I found very interesting, particularly how the better combination of immune response genes was giving humans and animals a more proficient and stronger immune system to fight diseases. And then my first postdoc after my PhD was in the United States. So I left the UK to go to Bethesda, Maryland at the NIH, where I had the privilege to study at the National Institute of Health, deafness and all genes involved in deafness, both in the mouse mutants as well as in humans. And I was fortunate to be able to publish out of the studies that I did in my postdoc 2 very influential papers in the field, which described 2 new genes responsible for deafness syndromes, both in humans and mice. And that time that I was at NIH, it was when Eric Green, now the director of the NHGRI, was setting up his first intramural sequencing lab. That changed really the way we used to do positional cloning to find genes for diseases in mice and humans, and sped up the whole process immensely through DNA sequencing. And this was absolutely remarkable, and I guess that I have been sort of a product of that revolution. Continue to really be interested by genomics and data and how we can apply those to health. I moved to the University of New Hampshire after my postdoc for a research professorship, but I also decided to follow an unconventional path, which is not the path of complete full professorship. So I was, I had a research professorship, and I was doing research, and I continued to do research on deafness, and I moved from the mouse model to the zebrafish model. And but I was also given the opportunity to be the manager of a new research facility in genomics, where they recruited me to, with my experience I had learned from the NIH, to apply all of the genomics understanding and building genomics tools for the application of those to economically important species. And it's here that I first started to work with different species, such as the cichlids of East Africa, the Daphnia prolix, and the strawberries. And this was also the time where NHGRI started to fund a 3 genome sequencing center in the United States, and when they had a white paper process where people and communities, scientific communities, came together to apply to have the genome sequence of a particular species done and so one of these centers was the Broad Institute of MIT and Harvard, and so I moved there first as a research scientist and remained there until 2014, where I left. And when I left, I was the assistant director of the genome of the mammalian genome sequence, vertebrate genome sequencing and analysis program. And this is the time of an immense genome revolution, where the new technologies and sequencing have allowed really to have costs of genomes decrease immensely and fast. We know the human genome costs are around $15 million, but now we are talking about $1,000 and $100 genomes. And this was also the time where the big data meeting biology, the 2 fields really came together and was really the time where a new generation of scientists were being trained to really ask different type of questions using this multidisciplinary expertise. And decoding the genome became what I was most interested in. And the more we learned about not only our own human genome, but also the genome of other species, the more we actually learned about ourselves, learned about the catalog of information that is extremely important for the functioning of life. And also, as I said before, an extremely important time where scientists like me really were brought together to combine a number of disciplines, and that is mathematics and computer science and statistical sciences and biology, and really understand the processes by which genetic variation, the processes of genetic variation and its consequences. So for me, during the time at the Broad Institute was very important time where we began to work on algorithms to assemble genomes, because of course the genomes, the full length of the DNA, the 3 billion letters, we could only begin to sequence with certain technology and in shorter fragments. And it was like a huge puzzle. And then we needed to reconstruct the original DNA sequence. And over the course of my career, I have been focusing on this problem from human genomes to vertebrate genomes, as well as the biggest plant genomes. And I have been developing and working towards the development of algorithms that have allowed the assembly of different type of genomes for different contexts and usefulness. And there's been a really close relationship between the molecular biology that I had learned in my degree and the computer and the statistical sciences that I then came to learn later in life. And this is the science of genomics, bringing together really the understanding of how to align sequence together, how to find motifs and patterns in the genome that are of relevance and function, haplotyping, the reconstruction of phylogenetic tree, the protein structure analysis, the genome assembly, the variant detection, the annotation, the elements discovery, gene expression. All of this has really be key and foundational to my research and my interest, even if the context has been somewhat changing at times, this being applied to environmental questions and other times to health questions. So the beginning for me started with at the Broad Institute, we really joined in a project which was after the first 4 genomes have been sequenced, the human, the mouse, and the rat, and the dog, and is comparing the genomes and understanding what can we learn by the sequencing of additional mammalian genomes. We already know that the genomes are like, we can read the genomes across the species like a Rosetta Stone. And we know that there are some parts of the genomes that are very conserved. And we know that if they are conserved, they are usually overlapped with very important and functional regions. So the question, I was able to participate in studies and that were very important and asked the questions such as how much of the genome is conserved under selection, how much can we detect, where is the conserved portion of located, and what is the features, the grammar of the that we can detect within our genome. So the genes, the non-coding portion of the genes, the promoters. And so we realized pretty much that if we looked across different mammalian genomes, in fact, as many as 29 mammalian genomes, and we published this in Nature in 2011, you could find that the portion of the genome that was evolutionary, the majority of the portion of the genome that is was evolutionary conserved lies in the non-coding portion, while the protein coding portion is just a very small 1.2%. So the most functional information is non-coded, and they carry most important elements that regulate the gene. And so we were able to detect conserved element across 4 mammals, but also understood that if you added more mammalian species, like 29, you were able to improve your resolution due to the phylogenetic distance. And discover many more conserved elements across the genome and understand at a much higher resolution. So you go from, with 4 mammalian genomes that you compare, you go to 50 base pairs, you go to 10 base pairs resolution with 20 mammalian genomes. And you begin to form a catalog of non-coding and functional elements that are indeed very important, because we know they are important now for traits. We know that over 90% of regions associated with disease and disease was are non-coding, and we know that conserved non-coding sequences are enriched for variants that are associated with traits and disorders. And of course, from there, we've known and we continue to know of the many huge projects that exist and that are trying to understand and decode these traits for diseases. By 2025, the total number of genomics data alone is expected to equal or exceed totals from 3 other major producers of large amounts of data: astronomy, YouTube, and Twitter. And we know that it's been huge, the current capacity at which we are generating data, and that we are absolutely being outpaced by this data generation in terms of being able to store it, being able to analyze it. We also know that we are moving, and I quite like these slides, because we are moving from the genome era to a social genome era as well, where we're now beginning to see a lot of consumers, company playing a really important role in whole genome sequencing and transacting in DNA markets and so on. So the big data market in healthcare is projected to be $78 billion by 2027. And yet, what we need to do globally in many countries is now progress to the second phase, which is the genomics in patient care, the adoption of genomics in the healthcare system, which in many countries and globally has required a system change and is requiring a system change. But one of the major issues is data sharing, and one of the major implementation issues. And this is one of the work that I'm doing right now with many consortia and on behalf also of Genome BC and Genome Canada, is trying to understand what are the challenges that are really stopping us from progressing the uptake of genomics into the healthcare system for precision medicine. There is a lot of challenges, many siloed data. Many of the funding approaches have had to adapt in recent times because they're not really been designed to cope with this majority of data. The security and legal and ethical issues, as well as the inclusion of equity in this genomic data and the underrepresented populations, the data infrastructure, which require huge maintenance and cost, and the need for data management. So this data management and integration is a big portion of what it's needed now, and it requires, it will require tremendous resources, but also it will revolutionize eventually medical therapies and personalized medicine. Many projects right now in the world globally are available for this, and we are learning from these projects. So they are what we call our learning systems. We have the All of Us project initiatives, where there's data from 1 million people, collected lifestyle, environment, health and disease, and some of these people have been given wearable sensors to collect additional data. The cancer moonshot in the US as well, reducing the risk of death by cancer of 50% in 25 years is the goal of this project. There is a cancer atlas of tumor change over time, as well as cancer research data commons. We have Genomics England, a great example right now from the UK, on how to uptake the genomics into a system change and into the healthcare. It's a $415 million initiative for 100,000 genomes, 100 rare diseases, 7 common cancers. Training and education are embedded, centralized infrastructures, 13 different centers for patient recruiting, research and industry access. Huge, exciting project from which we are learning, and a more recent project, Australian Genomics, that has received much government funding in more recently, and that I will be very excited to visit in October. And of course, there is also an explosion of genomics in private care, because because there is also industry has launched their own genome project. We've got Regeneron with the project, 250,000 participants using the UK Biobank, 500,000 exomes. So the importance here between the relationship and the link between academic and private industry, Foundation Medicine that are providing genomic solution for providers, for patients and industry, Fabrics Genomics. These are AI-based clinical decision support platforms for the interpretation of clinical whole genomes, but also custom assays. They're not just diagnostic, they're also clinical interpretations. It's very exciting. And I took the liberty, since and all of this learning system, the Global Alliance for Genomics and Health is learning how to navigate what we can learn, and how we can then take what we learn and how we adopt it for other emerging projects in other countries globally. So I think the Global Alliance for Genomics and Health is really important, and is definitely particularly concentrated around data standards, responsible sharing, interoperable exchange, informing policy, but also disseminate common practices from these learning systems. And here you have what I've learned today. I have decided to put this new slide here today for deciphering the Mexican genome. Now you have the origin, this very exciting new project for a biobank, and the first Mexican biobank. Unlocking the potential of health data requires engagement from multiple stakeholders. There is no doubt about this, that collaboration and engagement is absolutely key, including funders, including data providers, innovators, researchers from academics, but also private industry. And so there is really a need for open and efficient data sharing, alignment to newly developed funding mechanisms. So funders are a really important piece of the puzzle as well, and integrating very much with the private sector expertise. So having said a little bit about my interest in data and the sort of national efforts that I have been pushing in health, I want to tell you a little bit more, going back some in time, perhaps some of my early interest as a professor. And I applied, as I told you, I've been interested in genomics, and I've been interested in how genetic diversity and variation have contributed to traits, and how these traits have evolved, and how we can learn from it. And I have applied this to a number of different organisms and published a number of high-profile papers, and some of which are here. And I don't have the time to talk to you about all of them, but I wanted to just quickly talk about maybe the cyclic system really quickly, which is regulation, which is the understanding of regulation, and how that regulation has evolved over time, and how it has evolved also more over time, more than gene content. We know, in fact, that, chimp and humans, at the gene level, we are very, very, very similar. So most of the evolution of form must come from the non-coding portion of the genome and the regulatory portion of the genome. And I have decided to test this and really look at this in the model of East African cichlids. Now, I know that there are also Central and South American cichlids, but somehow they have not evolved the wonderful traits, the plethora of traits and that you see in East Africa, where they've undergone massive radiation and explosive radiation, and they are known to be textbooks radiation. So you can see some of the differences in colors. You can see some of the different in morphology. There's differences in behavior, the differences in feeding behavior. And when I was at the Broad Institute, there was a very big project that I led with the scientific community, and we sequenced 5 genomes from the different radiations in the 3 different East African lakes, one from Tanganyika, Malawi, and Victoria. And we looked at the genomes of these cichlids and compared the genomes with other vertebrates and other teleost genomes and started to ask the question, is there anything in the genome that can tell us what makes this fish really good at adapting and are expanding and are diversifying? And we found that indeed, I can't go into many of the details, but we found that protein coding changes were extremely important, in particularly for the development of coloration and different colors. Transposable elements played a huge role in the evolution of new promoters regions. Gene duplication were extremely important, as well as microRNAs. And I give you we found some very important genes such as BMP4 and NOG2 and BMPR1B, which are all acting in the same pathway, very important for morphology, as well as protein coding evolution in the endothelium genes, which we know in fish and mammals cause color, but in mammals also tend to cause Hirschsprung syndrome and the Pied-Baldita in mouse. So anyway, at some point while I was doing all this great work on many, many genomes, I decided to move and take this opportunity to go back to the UK. And I went to the UK where I decided to take this position as the Director of Science at the Erlam Institute. This is a data science institute reporting to the government. And here I continue to really work by bringing together this data with biological skills. And I worked mostly on developing algorithm. I moved from assembling genome and analyzing the content of the genome and the non-conserved element of a genome. I moved to understanding transcriptions and expressions and how the expression levels, we could reconstruct regulatory networks, particularly using the transcription factor and using the knowledge that I learned previously about the non-coding portion of the gene. And so I'll go a little fast. I applied this to many, many data, transcription data that we generated from the cichlids and reconstructed the regulatory network that were tissue specific. We collected data from various tissues like the gills and the brain and the liver. And we were able to really associate particular genetic variation with the evolution of certain traits and certain specific network. And we were able to also identify episodes of network rewiring that were extremely important in the rapid diversification of the traits in these cichlids. And so that is one story. And while I was a Director of Science at the Erlam Institute, I got involved with the UK government because the UK government around 2016 started a big push about global challenges. And they were giving a lot of money for global challenge research funds. And this is how my work in international development started. I asked myself a question: could I use my genomics and my knowledge of genomes to try and understand how I can improve major threats, which are not only diseases and health, but also climate change, biodiversity loss. And it is with a very small amount of money, I think it was about £30,000, that I initially created a network. I was given some money to create a partnership between UK and Colombia, and we called this partnership Bridge Colombia. This partnership is still active today, and it has grown enormously. Please visit bridgecolombia.org if you want to understand more. And we spelled bridge, the same letters, both in English and in Spanish, spell the same words of biodiversity, responsibility, innovation, development, growth, and education. And I'm just gonna show you a very, do I have time? How much do I have? I'm gonna skip this then. Okay. I'm gonna skip this. You can go. I'm gonna tell you this has become a very big, network. It involves over 50 research organizations between UK and Colombia, industry, NGOs, and governmental agency. And it promotes the sustainable use of biological resources through a process of genomic responsible innovation. What I want to tell you is that the initial 30,000 pound network that we put together was so successful that we then were able to secure a number of funding under the Global Challenge Research Fund in the UK. And that was for one project called Grow Colombia, which was a £6 million project to do research on biodiversity across the UK and Colombia. And I'm going to tell you a little bit about this, but do please, we have a website. It's called growcolombia.org. Please do go and check the website. We have, as part of this project, I was able to also join the Misión de Sabio. I worked with and I continue to work with the 3 different administrations in Colombia, Santos, Duque, and now with the newest administration just now, advising them on science, innovation, and technology. And we were able to create this network that I told you, Bridge Colombia, but also with the £6 million, start a project that lasted 5.5 years and that has had a tremendous impact in capacity building, in understanding natural diversity and collaborating with the major research institutes in Colombia to advance genomics for the understanding of species, agricultural diversity for the understanding of how to improve economically important species for food security and for more sustainability, and also a socioeconomic program, which is understanding how to also use all the tools of research to give back to community, underrepresented community, but also promote social growth. And lastly, I want to tell you about this other project that we started, which is the Colombian Earth Biogenome Project, which is the Colombian project. The Earth Biogenome Project is a global initiative. It has membership all over globally. And Colombia has joined this global initiative for a sequencing effort as part of our project of different species. We had many partners and collaborators and funders over the 6 years. Many people, postdoc students, PIs across the UK and across Colombia, too many to put in one slide. This is the reach of our projects across Colombia. These were all genomics projects in biodiversity, and mostly was to prevent biodiversity loss, to accelerate breeding and sustainable farming, and to strengthen policies. I just want to really, I'm finishing, and I just want to tell you this project yielded 44 papers across the 2 countries, many databases, many research programs, 2 policy briefs that went to both the UK and the Colombian government. We hosted the 35 capacity building events, mostly all in Colombia, engaging more than 1,100 people. Public engagement events, we have a web page and a YouTube channel. So just go to Google and search Grow Colombia YouTube or growcolombia.org, and you will see the stretch of this. We have also published a book through a competition for early career and professional writers. We selected 12 of them, and they wrote stories about our research, our genomic research. And the book was presented at COP. We were invited at COP26 this year to present our book and our story. We also engaged schools and youngs. So we did our science and then we took it to the schools. And again, one of our school projects called the Living Maps, we made a little film, and this film was showcased at COP26, and we involved 56 teachers, 400 children, and 38 underrepresented schools in Bogota, Medellin, and Cali. And then lastly, one of the programs that also we did with genomics is to train underrepresented population and rehabilitate. And this is a program that continues to be funded by many different sources and has had a lot of success. It's led by Jaime Gongora at the University of Sydney and involves many partnerships, but we were able to put the 16 ETCRs in Colombia and representatives from each one of them together and trained the ex-combatants and rehabilitate them to become guardians of the territories and sampling the biodiversity and being able to teach them how to sample our biodiversity for our genomic studies. And this took about 3 years preparation. And 100 flora and animal species have been documented since we started this program, over 100, a biodiversity booklet for an ecotourism route, developed a 9 naturalist project. It's been really great. I'm not going to show it to you now, but there is a video on the YouTube channel. So if you want to see some snippets about these projects, and I'm just going to end by saying thank you and happy to answer any questions.
Great presentation, Doctora Di Palma. Actually, it's inspiring. It's very creative and very interesting. I think with your help, we can go for growing Mexico, the next project, hopefully with you. We need to do this because we have also a lot of diversity that we are not considering in our research. And also one of my comments is because I am, at least me, is my personal opinion, we are emerging in the clinical aspect of genetic and genomics. And all diversity that you are showing is very interesting because it's the interaction with the human being and all the ecosystem. And we should consider it in our research too. Thank you very much for this presentation. And here, Dr. Treviño has a specific question. It's about, now that you know our projects, for example, Origin, how do you see Mexico, or at least our institution tech, how we can contribute to the global genomics as with your eyes, with your expertise?
Well, I think, first of all, this visit has been really great. I mean, the campus and the facility that you have here are truly impressive. And I think that is your strength. I think that starting a biobank for Mexico is a fantastic idea, is necessary. We know from all of the projects that we have right now globally from the Western countries, we know we have one huge problem in health genomics, and that is the lack of diversity in our cohorts. It is extremely, projects from Mexico, such as the one Origin, countries like Mexico are key really to our future of understanding health and disease much better, are key to the genomic equity that needs to be achieved. So I think these are absolutely important projects, and they are going to have a massive impact in the global scale. I think that one of the things that has been very difficult with me working in middle income countries and sometimes is the lack of capacity within the country to be able to generate the data. But you have a core facility, you have a NovaSeq. I believe that I've learned that this is the third in Latin America. So this is really important and puts you in a very different position. It puts you in a position to really follow through from the biobank, utilizing the biobank. The biobank itself is going to be useful, on tremendous use on a number of but it's also going to be useful for augmenting your opportunity for private partnerships, for example, I think. And so how do I see for Mexico to contribute to the global? I think by doing all the things that you have been doing, but perhaps by engaging collaboration internationally a little bit more and acquiring some visibility for the project and early collaboration. Like I mentioned in my talk, the Alliance for Health, the Global Alliance for Health. You already connected to the UK Biobank, but just sort of understanding how collaboration and multiple stakeholder engagement can come to move the project forward, I think will be important.
Excellent, thank you. This project is just in the first, in the beginning, so probably with the time, people like you will start knowing about our project, and probably we start sharing with other people and other company about at least that people outside from Mexico that know the project that we are running. And there is a second question about, we are doing biobanks for human samples. Do you think, or do you consider that in parallel, we should start doing our biorepository about environmental and species and plants, another type of biobank, or is not necessary, it's just doing research? What do you think about?
I think absolutely. I think this is the reason why in my talk, I had to go very fast between health and environment, because I had to show you the possibility. I wanted to show you and talk about this. So I've been very involved with the Earth Biogenome Project. There is exactly that. It's about the sequencing of all of the biodiversity in a country for social and economic growth. Now, Mexico is in the same way. For Colombia, I've started a project in Colombia exactly for this. We partnered with the government, who, as you know, under President Santos and under the Green Growth Strategy, started many expeditions in the territories to sample biodiversity that has been previously unknown and to increase their cataloging of biodiversity. We paired up, with this effort to try and understand how we could then take this further to the sequencing of some of these biodiversity. And obviously, Colombia is a mega diverse country. Mexico is mega diverse. It will be very important in this case, in the case of these countries, that there needs to be a prioritization. So you can't just sequence the whole biodiversity, it's impossible, but will have to be a prioritization. So different countries will prioritize. Colombia is prioritizing clusters where the diversity mostly gives economic return, like, so, products, new medicine, new products, something that can be commercialized. I think for Mexico should have a project as well, should have a Mexican Earth Biogenome project for sure. The Wellcome Trust in the UK has just made a pledge of several million pounds to sequence the entire diversity of the UK. It's only 66,000 species, because there is no diversity in the UK. So I think about what they are doing, which is remarkable, is they are now working with the Global Earth Biogenome Project, of which I am the international scientific chair, to develop all of the processes, the methods, the data issues, the regulatory issues, the methodological issues. And so they are leaving a huge they are leaving the opportunity for other countries to design a project like this, and they don't have to start from nothing.
Excellent.
So yes is the answer. And you have your core genomic facility, which can be developed on this.
Excellent. So great. Thank you very much for this information, Doctor. You inspire us and give us many, many ideas to work with you and to do many things together. So I give the voice to Arturo. Arturo, you continue with this session. Thank you very much for this presentation, Doctora Di Palma.
Thank you. It was a pleasure. Thank you very much.
Thanks a lot. So I would like to thank Federica Di Palma for this amazing presentation. It was such an interesting topic for us. And also I would like to say thanks to Dr. Rocio Ortiz for the kind moderation of this session. Now, next for me, it's a great pleasure to introduce Dr. Ángeles Domínguez, Associate Dean of Faculty Development, that she will share with us a little bit of some of the information about the speakers of the 5th season of the Cátedra Magistral. So welcome, Dr. Domínguez.
Thank you so much, Dr. González. This is my pleasure, first of all, to thank Dr. Federica Di Palma for the great talk, as well as to Rocio Ortiz for the kind moderation. We are going to present to you the next 4 sessions that we have for the 5th season of the Cátedra Magistral. So on September 14, we're going to have Dr. Eduardo Carrillo Maravilla on Wednesday, September 14. And then we have Luis René González on October 5th. Then we have in November, Dr. Rocio Ortiz, that she will be now our invited speaker. And we're going to close the session, the season, with Dr. Luis Arturo Alonso Herrera on the last day of November. So this will be our season for this year, this 2022. And we thank everyone for letting us the date and of course sharing with us the events. Thank you.
Thank you, Dr. Domínguez. Well, this is how we conclude the first session of the 5th season of Cátedra Magistral Profesores Destacados. On behalf of Tecnológico de Monterrey School of Medicine and Health Sciences, we thank the audience for its participation. We emphasize our appreciation to our invited speakers, Dr. Federica Di Palma, for her wonderful and outstanding presentation. We also thank Dr. Rocio Ortiz Lopez for her support as moderator of this session. And last but not least, we extend our appreciation to the organizing team that make this possible. Now, please remember that you can find our session at YouTube in the channel of Tech Salud, and also we invite to all of you to the next session of the 5th season of the Cátedra Magistral, Profesores Destacados. Depending on our social media networks, we leave you with a video of our school. Take care and have a wonderful day.
Hay eventos en la vida que nos cambian y dejan una huella en nuestro interior para siempre. Momentos donde simplemente todo hace clic. Algo en tu interior llamaba y estabas listo para responder. Una explosión interna, el nacimiento de tu vocación. Sentiste emoción y un poco de miedo, pero también determinación y responsabilidad. Cambiaste. Empezaste a ver esperanza en situaciones de desesperación, a tener templanza en circunstancias difíciles, a escuchar a quien más lo necesitaba, a investigar cuando no se veían soluciones y preocuparte por cada aspecto de la salud de una persona. Ahora llegó tu momento de unirte a nuestra comunidad que te recibe con los brazos abiertos, listos para acompañar tu intuición, tu llamado, tu vocación. Tech Salud.